Our moderator for this session, Dr. Arvind Sahu, has completed his PhD from Vallabhbhai Patel Chest Institute, University of Delhi. His postdoctoral training includes positions at University of Pennsylvania and University of Texas Health Science Center at Tyler. He is a recipient of many prestigious fellowships and also has many patents to his name. Sir, I request you to kindly come to the dais and chair this session. Our first speaker is Dr. Sunil Arora, a renowned name in this field. I request the moderator to briefly introduce the speaker. Thank you.
Thank you. So welcome back everyone again, and we have two exciting talks in this session. The first one is by Dr. Sunil Arora. He's professor in immunology in Pigmer for long time. I think he started his individual career there and then strayed, and everyone in the immunology fraternity knows him. We have a little community where we meet every year called Molecular Immunology Forum, and therefore we come and informally discuss our work, and Sunil has been part of that for a very long time, and that's how I came to know him and his work. He works in diverse areas, but immunology of infectious diseases is one of them, which is his main forte, and also he works on stem cells and other and cancer as well. So he kind of covers a large area. Excellent scientist and excellent human being. A very good friend of mine. So Sunil, please, it's all yours.
So good afternoon, and thank you very much, Dr. Arvind Sahu, for a nice introduction. And I would like to thank Dr. Sharad Dalai for inviting me here to this CME. So essentially, we all came here with the mindset of teaching the students who are not very much exposed to immunology. So as Arvind said, my lab has been working on many areas, specifically immunology of infectious diseases, and that among them, some of them are listed here. So, but I am not going to talk about all these today, so I will restrict to teaching, and my lecture would basically comprise of antigen presenting cells, antigen capture, processing, presentation, and activation of T cells. And I will end my talk with the last slide where I will have connection with Amit's lecture, who is going to talk about T cell polarization. So I say that most of pictures and some of these, tables or whatever I'm going to show, they have been taken from different books and different, literature. So I don't claim them to be mine. It is purely for teaching purpose. And I'm happy to be here, and I have really enjoyed sitting here and seeing the interaction of students. Many of the students are really good. They ask very good questions. So let us see how much you remember. Since yesterday, we have been talking about this immunology and activation of immune response. So let's see. So this is basically, in a nutshell, we all, you can say that this is how the immune system gets activated once an antigen is seen, right? So it has been said that antigen enters into the body and triggers the immune response. So what are the first cells which need to see the antigen?
Neutrophils.
Excuse me, sir.
I am also going to ask him.
dendritic cells.
ला दो इसको हाँ good good thank you sorry okay so here in this picture what is the color of that cell which will look at the antigen can you recognize that cell one हाँ यार कलर क्या है उसका yellow very good so this is the cell and the antigen is here red color so red color antigen has come in and the macrophage or maybe any cell which is phagocytic in nature and that is the in general we have been learning it right in the school science that there are cells which have the capacity to engulf anything which comes in your body, try to degrade it and get rid of it. That is what the यार Amit, please don't laugh. He makes me laugh. इसको उठाओ यार. Sir, chairperson, I object to his sitting. अच्छा तू उधर मुड़ करके बैठ जा. Please. I'm sorry. You can sit here. Don't worry. Okay. So the red cell, red antigen entered into this. So it's trying to do that. And once it does that, it gets activated. It releases certain cytokines, soluble factors, which activate other cells around it. And it also attracts many more cells to the site of infection. And those all cells, they cause inflammatory response. So initial inflammation occurs and then you have heard about innate immune response and then all these cells which are the components of innate immune response. Yesterday Dr. Dipyaman Ganguly and then one after everybody is talking about innate immune response and then adaptive immune response. So you have all these components, they generate a innate immune response and madam also deliberated on that innate immune response doesn't need very specific receptors but there are general receptors and what are those receptors? PAMPs are what? PRR. So they are the PRR, pattern recognition receptors, and they look at PAMPs, right? Or death associated or damage associated. So all those things they happen and they activate and when NK cells come here, neutrophil come here, they all take part in that part. But this this particular cell, which has actually eaten up the foreign which is pathogen or whatever. So its role is not over. He is not satisfied with just that. So it will degrade something and then it will activate another cell here and which is a CD4 positive cell. And once that happens, see there is a connection between, they are talking to each other and this cell becomes, this is what is antigen presentation. And this cell becomes activated and starts, releasing more cytokines and talking to all other cells and then activation of all these cells, which is called the adaptive immune response, and there are specific antibodies formation. And so in nutshell, we know about this. But there is more into it because when this cell has taken up, so there are two things which we remember. What is innate immune response? We already said it is not very specific. There are some receptors which are called pattern recognition receptors. They look at PAMPs and all this, and they look at initially the microbes as well as the injured tissue and dead cells. So both of them they activate. So this is limited with inflammation and antiviral state and stimulate further the adaptive immunity. And what are the features of adaptive immunity now? Because it has been said by all since yesterday that there is a specificity. Madam also elaborated on so much of diversity, and ultimately the aim is to create some kind of specificity, right? So what is that specificity is the major feature. It also has the memory, right? Which memory is missing in the innate immune response. Lot of diversity, and that's what she taught. I mean, nobody else, I think, can do it in such a good manner in just 40 minutes, because it's such a complex. We all get really bugged by the various combination, permutation, and ultimately coming out with the T cell receptor or a B cell receptor. So there is a lot of diversity required in the adaptive immune response. And then there is a self, non-self discrimination, which is also a part of it. So these all 4 features of adaptive immune response have to be there. So to have all these things in the adaptive immune response, so the antigens will have to be captured, processed, and presented onto some molecules which are the MHC molecules, because they are the ones which basically responsible for recognizing self, non-self, right? Everybody said. So there has to be something. So all those antigen, those yellow color cell which I showed, somebody said B cell, somebody got dendritic cell. So these cells, which are called antigen presenting cell, which talk to the naive T cell or the T cell which needs to be activated for generating specificity, have to have all these qualities to become. So second thing is that if the T cell has to be activated in an antigen specific manner, then the T cell has to go and find the antigen, right? But that part is done by the antigen presenting cell. And because there are very few lymphocytes in the body which have specific for one microbe, and because of the diversity which is created, so that is possible. And there are few lymphocytes must be able to locate the microbe, and these are all done by that they express certain chemokine receptors and move to the site. So the antigen presenting cell which has captured the antigen has to move from the site of infection to a place where the naive T cells or the T cells, they come and interact with it. So both ways it is important that they should have the capability to move and migrate and come together, and only then they can see each other. So therefore antigens and lymphocytes have to be brought together, and that function is done by lymphoid organs, right? Okay, so the sites of antigen entry, most of the times skin, gastrointestinal tract, respiratory tract, and the sites of antigen capture are naturally there. But which cells they will do? They are the antigen presenting cell. They are the phagocytic cells. Let us not call them at the moment antigen presenting cell. So they are the cells which will capture and they will try to get rid of it. But they have the capacity to migrate, go into the lymphatic vessels. And if the antigen happens to be in the bloodstream, then it goes into the venules. And these cells, they come to the lymph node, where all other blood antigens, they go to the spleen. And that's where they want the T cell to come and look at the antigen. So they don't go to the T cell areas. So that's how the antigen which has been captured from all these areas, they are brought to a place where there is a likelihood of the lymphocyte to come and see that antigen. So that's where basically because the T cells which are maturing in the thymus, they also express the same chemokine receptor as the antigen presenting cell or dendritic cell, which expresses CCR7. That's what Dr. Deepaman Ganguly said. So both of them, they express same chemokine receptor, and that's why they move to the same place, because the chemokines are here. So that brings both the cells, this antigen presenting cell and the T cell here, where they meet and they do it. So the sites of microbe entry, the skin, GI tract, airways, organs of continuous epithelia and populated with dendritic cells. These are the cells where most of the dendritic cells or antigen presenting cells are roaming around, and they are less colonized in the tissue and blood. But the sites of lymphocyte activation are the lymph nodes, lymphoid organs, the lymph nodes and the spleen, and the mucosal and cutaneous lymphoid organ. So the antigen presenting cell carry the antigen there, and the T cells, they go there and they meet and that. Okay, so far so good. That's what we have learned and everything good. So antigen and T cells come together and in the same organ. So now comes the question, what are the characteristics of a good antigen presenting cell? Because it has been said very clearly that the T cells, they do not look at the antigen which is in soluble form. It has to be presented to them nicely. So they are very nakrewala, like Amit. You know, he will not look at anything unless it is presented to him on a properly, on a, you know, a cup of tea should be on a tray, and somebody should come there and present it to him. So he will take it, otherwise he rejects it. He doesn't want to look at it. So that kind of T cells, they don't look at it unless it is presented in a proper manner, right? So that proper manner is done by, has to be done by some cells. Somebody has to do. The B cells are more open, you know, like me. They will look at antigen while they're roaming around, you know, in the soluble form or whatever. So they are cool, you know. He's a tough guy, right? So these T cells are tough guys, so they have to be handled carefully. That's how the antigen has. So let us see what are the properties of a good antigen presenting cell. Has to capture the antigen, that is prerequisite, right? Has to process the antigen in a way that the whole organism or whole bug cannot be put on the MHC to be presented, right? So it has to process the antigen and put something on the MHC, which has to be loaded. So it has to, the cell, which is going to be a good antigen presenting cell, should have all that machinery and then present in conjunction with the MHC molecule. Only then the T cell will look at it. Right? So, and then only the T cell will get activated. So all these prerequisites we have to see, and that's why we call some cells as antigen presenting cells, because they have all that machinery with them. And the prerequisite for them is that although they can capture antigen, many cells can capture antigen, they may not have capacity to degrade it and process it, so processing defect, or they may have problem of not expressing the proper MHC molecule, and so loading defect or something. So there are definitions now. So antigen processing has become a very important phenomena to generate or active the immune response. So what is antigen processing? It's basically the big proteins, the peptides are cleaved and processed, and then to a specific size so that they can load it onto the MHC class, MHC molecules. Let us not say class 1 or 2 for the time being. So these MHC molecules, once they are there, so we have to have a proper tray. So if it is like this, the cup has to come here, so it should accommodate that cup. Otherwise, you know, Amit will not take the tea. So that tray has to be, can accommodate a small, you know, nice cup and saucer. So if it is bigger or, you know, distorted, it will not hold it. So that's why. So the presentation, that antigen, once it is there, so it has to be loaded onto the MHC, which will see that it's of appropriate size and specificity, so it will bind to it, and then it will move out. So that these are two definitions which we need to remember, that processing means breaking off antigen into smaller peptides and then loading onto the MHC to make it MHC peptide complex, which is presented outside on the cell surface. Okay? Good. So for that, you need to know what is MHC, and it has been told that there are MHC class 1 molecules, which are there on all nucleated cells. So from that angle, all nucleated cells should be able to present antigen through MHC class 1. The features of MHC class 1 are that it has a alpha chain associated with the beta 2 microglobulin, and this has got a domain in the alpha 3 which binds to CD8. That you should remember for the time being. Let us not, I will show you where the peptide comes from, but this is class 1 and it has got this kind of feature where this is a transmembrane, this is not transmembrane. This is non-covalently associated with it, and this stable molecule can bind to CD8. So obviously, whenever there is MHC class 1 which is carrying the antigen, it should interact with CD8 cell, right? Because it has a binding of CD8 molecule binds to it. CD4 doesn't bind to it. That means, right? And the class 2 molecules, they are mainly expressed on some cells, not all nucleated cells, and all these cells basically have the capacity to phagocytose and degrade and so all those things. And there are some cells which can be induced to express MHC class 1. So there are some which are constitutively expressing. They are all good. They are the cells which can present antigen through MHC class 2. And MHC class 2 has got 2 chains, alpha chain, beta chain. Both are transmembrane. Both have 2 domains, alpha 1, alpha 2, and beta 1, beta 2. And alpha 2, beta 2, they are anchored, and this is outside, and which makes the binding of the peptide. But the difference here is another one, which is that it can bind to CD4. So this domain binds to CD4 molecule. That means all antigens which are presented through MHC class 2 will automatically interact with CD4 cell. ठीक है ना? Because CD4 makes a co-receptor. It goes and binds to that. And CD8 doesn't bind with MHC class. So there is a demarcation, and this demarcation basically leads to antigen presentation to different kind of cells and generate different effector cells which further differentiate, which Dr. Amit Avasthi will show you, but I'll just touch upon them. So both of these MHC molecules, they have the capacity to accommodate peptides, whereas alpha 1 and alpha 2 has a cleft which can accommodate a peptide, linear peptide, which is 8 to 9 or 10, 11 amino acids. So this can be accommodated here, where the binding site is shared by alpha 1 and alpha 2 of the alpha chain only, right? Beta 2 microglobulin doesn't bind with doesn't take part in the antigen binding. Whereas in this, both chains, they take part, because alpha 1 of alpha chain, beta 1 of, both of them, they form this kind of cleft where the antigen can be loaded, and both, they participate in that. Good? Okay. So all MHC molecules have similar basic structure. The cleft at N-terminal region binds peptide antigen and is recognized by the T cell receptors, and the membrane proximal, they have the capacity either to interact with CD4 or to CD8, not together, right? So once the antigen has been loaded, I'll tell you that part a little bit later, how it is loaded. But what the T cells look at is both here. This is the T cell receptor. This is the MHC with the peptide. So the T cell receptor recognizes the peptide because this T cell receptor, as Dr. Vanita Bal has shown, that there's a lot of diversity and there is one T cell receptor which is specific for one antigen which is presented here, and that binds, but it has to look at MHC molecule also as a self-form. That's what I asked her, what is, MHC, this is MHC restriction, that the antigen is presented by self-MHC, is seen by the T cell receptor. So the recognition comes in by recognizing the peptide as well as the MHC. So peptide has to be foreign, whereas the MHC has to be self, right? So that is another question, we will deal with it, that if they say kidney which is transplanted, all kidney cells, they have MHC class 1 or whatever. So they are presented and they are, the immune system gets activated, and though they are not presented, but our T cells, they recognize that as foreign, So that is a separate question, we'll deal it later on. So T cells can recognize only cell-associated protein antigen and display on the MHC molecule, right? So there is something more into it. It was yesterday also told that the antigen presenting cell present the antigen through MHC because it has acquired the pathogen, degraded it, and loaded it onto the array, and TCR reacts to it. But this covers only one signal 1. But when it looks at the pathogen-associated molecular patterns, which are seen by PRRs, which are TLR, NLR, or those receptors, one of those receptors, they basically help them to upregulate some more molecules. And those molecules, they pass on the co-stimulatory signal, which is absolutely important to generate a proper, signal 1 and signal 2 for activation of this knife cell. Although there is a third signal also, which basically has to be dealt by Dr. Ahmed, that how the T cell after activation, after seeing the antigen, recognizing it, it will polarize further to form an effector cell, so that this signal works here to decide which particular lineage it will become and how they will further become a functional state. Okay? So this all is important, but then the question still remains, this pathogen has been taken up, this only arrow shows that it goes there, it is not that straightforward. There is a proper process which happens, but the antigen presenting cell and the T cell, besides TCR, has got so many other molecules, they also bind to each other and they hold it together for the signals to pass from the one cell to the other. And as you see here, the CD4 acts as a co-receptor. So don't confuse CD4 and CD8 as a co-stimulatory molecule. So co-stimulation comes from one of these molecules, one of these signals, right? This is a co-receptor because it doesn't have any counterpart here, but it binds to the class 2 molecule, and CD8 binds to the class 1 molecule, okay? So when all these properties are together, we find that these are the 3 type of cells which carry all these properties. They have the capacity to take up the antigen, process it, load it onto the MHC molecules, and then express it outside, right? So out of these, dendritic cell is the most potent, as it was told to us yesterday, and we all agree to that because they have a extra capability, which is that they are present, and so they pick up and migrate to the lymph nodes and then present the antigen, and during migration they mature, and during maturation they express a lot of co-stimulatory molecules, and co-stimulatory molecule pass on second signal, and they are good antigen presenting cell, and they are the cells which present the antigen to the naive T cells. Those cells which have never seen the antigen, they are the ones specialized cells which are, whereas macrophages, B cells, they also do the same. So this is what dendritic cells do. Normally they are immature, but once they see PAM, they take up the antigen and process the antigen, and in that process they migrate and become mature cells. So there is a clear-cut phenotypic difference between immature and mature dendritic cells. You can actually plan experiments looking at whether the cells are good or not antigen presenting cells, because in certain disease conditions, the pathogen or tumor, they down-modulate the capacity, maturation capacity of these immature dendritic cells. So if they don't mature, they basically do not have all that, co-stimulatory molecules expressed, so they remain poor antigen presenting cells. And in that condition, they really pass on a tolerogenic signal rather than a activation signal. So these are, some of the ways these pathogens or tumors, they evade the immune response. They make the antigen presenting cell defective. So these are macrophages. They also can take up antigen, process it, degrade it, and load it onto this center. Similarly, B cells, as madam said, they can look at the globular epitopes or soluble antigen. There is a big peptide. On that, there is a B cell epitope, which is totally different from a T cell epitope, because T cell epitopes are more or less linear. They may not be linear. B cell can look at even a conformational epitope and still bind to it, and then engulf the whole protein, degrade it, and then present that to the T cell. But then that epitope will be different, but the T cell epitope may be associated very closely on the same chain, and that T cell epitope is presented to the T cell, and the T cell can get activated. So we have these professional antigen presenting cells, the dendritic cell, macrophages, and B cells. They have all those features of good antigen presentation. And then we also have some cells which can behave as antigen presenting cells. They are called atypical. Under certain circumstances, they also express MSC class 2, and they can present the antigen. But more or less, most of the times we'll have the. So now, to make a antigen into a peptide, linear peptide of a specific size, there has to be a processing mechanism, and there are well-placed processing mechanisms depending on what kind of antigen the cell is handling, right? If it is a viral antigen or a tumor antigen, which are basically, the proteins are expressed endogenously in the cytosol, then all those endogenous proteins are processed in cytosol, and most often they are expressed, they are loaded onto the class 1 MHC and presented to the T cell. And as I told you, all those antigens which are presented on MHC class 1, because they can interact with CD8, so they present it to CD8. So that's why CD8 cells are more important for viral infections and tumors, right? Whereas all antigens which are acquired exogenously, like bacteria or other things, big proteins, they are phagocytosis, endocytosis, they are taken up. And we all know that whenever an antigen is taken up by phagocytosis or endocytosis, they fuse with lysosome. There is a lysosomal phagosome, and all those lysosomal enzymes, they break up and degrade it, and then all this process happens. So there are two different, clear-cut, different pathways of antigen processing, the endogenous proteins and the exogenous proteins. And all exogenous proteins normally are presented on MHC class 2, and they activate the CD8 cells because MHC class 2 binds to CD4, sorry. So, okay. I get confused when I look at him. So he sometimes makes some gestures. I forget my line. So instead of CD8, CD4, so never mind. But you guys are with me because you are understanding right? So there's no problem as long as. So let us see a little bit more of this. So endogenous antigens are derived from proteins produced into cell, and these include altered cell proteins, non-cell proteins, endogenous antigens associated with MHC class 1 molecules, we all cell, right? So processing of antigen, assembly, and then transport. So how do the proteins get degraded? They are expressed inside. So the phenomena is that ubiquitination. You all know that there is a process of proteasomal degradation of proteins. They are all those proteins which are not wanted by the cell. They are all proteins which are they are not folded properly, or they have become redundant. So cell will like to get rid of it. So there is a very good machinery which is called proteasomal degradation. All those cells, all those peptides which have to pass through this kind of tunnel, right? You all know that proteasomal tunnel. So this carries a lot of enzymes, the chymotrypsin, trypsin, caspase-like enzymes. They all have capacity to chop the proteins. So the proteins which have to pass through it have to be ubiquitinated. So this is ubiquitination reaction, which is energy consuming reaction. So all those ubiquitinated will come here. Ubiquitin is removed, but this goes through it religiously and chopped off. And many times they are converted into amino acids, so which are the raw material for, so they are being reused like for making more peptide proteins. So that's what happens with all these endogenous antigens, and these are peptides which are generated, and some of these peptides are really of the size which can be loaded onto the MHC class 1. So not all, but some of them, right? Because it's not so controlled, it will try to chop off proteins and degrade them and make them, okay, right? So all those peptides which are basically of the appropriate size, they are transported from the cytosol to the endoplasmic reticulum through these two transport associated of peptides. So these are TAP1 and TAP2. They help these peptide, those small peptide bits to the endoplasmic reticulum. And in the meanwhile, the MHC class 1 is getting arranged. So MSC class 1 has got the alpha chain, as I told you. So initially it is single chain, but it is stabilized as it binds to certain chaperone proteins, the calnexin, which binds to it and carries on. Then whenever the beta-2 microglobulin comes in, so this process occurs, and the tapesin and the calreticulin replaces the calnexin, and the beta-2 microglobulin gets attached to it. So it forms a protein, the MHC class 1, but is still bound with these two proteins. These are chaperone proteins. They stabilize till the peptide of the right size can be loaded onto it. So the peptide comes here, it loads, and then these two are released. And you have this peptide loaded MHC complex, which is pushed out on the membrane. And you have the MHC class 1 loaded with antigen, right? So this is summary of this process, and you have the MHC class 1. So MHC class 2 loading is a bit different from here, not bit, totally different, because it happens for the exogenous antigens. The exogenous antigens are derived from proteins produced outside the cell. They can be all these pathogens which are derived from outside the body, and these are loaded onto the MHC class 2, and then. So these are 3 steps again. So the difference comes here, that there we have the MHC class 1, which is stabilized with the chaperone proteins. Here the MSC class 2 is stabilized with another invariant chain, which is the linear chain, and then is degraded, and you have the clip. So the peptides bound to MSC class 1 are derived from engulfed pathogens. Macrophages internalize it by phagocytosis as well as endocytosis, whereas dendritic cell and B cells, they can take up antigens by endocytosis also into the endosomes. So exogenous are degraded, and then they are made of somewhere 13 to 18 amino acid long and appropriate size which can be accommodated, and then they are taken up, loaded onto the MSC class 1. So this invariant chain keeps binding to it till you have reached a stage when the phagosome, lysosome vesicle carrying the peptides fuses with this endosome, and because of the acidic atmosphere, the cathepsin, some of the enzymes, they start chewing this invariant chain to make it a shorter one. So this is what is invariant chain, and then when it fuses, the cathepsin S and L, it cuts it, and only a small portion which is called clip, which is class 2 associated invariant chain peptide, which remains bound to MSC class 2 antigen binding groove till a peptide of the appropriate size and specificity comes and can be accommodated. So the peptide comes from where? There is the peptides are engulfed in the endosomes, which fuses with the lysosome, endosome-lysosome fusion, it degrades, and then that fuses with this, and then you have C2V vesicles, which carry the class 2 MSC plus peptides, but then since clip is attached, the peptides are not getting loaded. So here comes the endosomal HLA, which is expressed only on the endosomal membrane. They are the HLA-DM, which binds with it and releases the clip, and the peptide gets loaded onto it, and you have the MSC class 2 loaded with the protein. So this is the summary. So this is lysosomal. So many pathogens, they manipulate with antigen presentation. Some of them, they block this, some of them, they block whatever pathways and try to evade the immune response. So this is how the peptides are generated, and then they get loaded and they are expressed outside. Okay? So this is the difference between the exogenous pathway, endogenous pathway, and we already delved. I think you all are clear how the two are different and why we have more of CD8 cell when there is MHC class 1 loaded, there is CD4 cell when the class 2. Okay? Very clear cut, and here are the differences and all, right? So this is again a summary. This is endogenous, and that we should not. So there is always the rules are there, but then there are exceptions. So there is a phenomena called cross-presentation of those antigens which are expressed, and they are taken up exogenously but are presented by MSC class 1, which is called cross-presentation, and dendritic cells are the best ones, but it happens in tumors or viral infections. And the primary and the most efficient are the dendritic cells, but sometimes people have shown experimentally the B cells and other cells, they also cross-present the antigen. So this is the one, the endocytosed antigens normally will go here, go through this machinery, and will be expressed by MSC class 1. And if it is the cytosolic protein will go here and expressed MSC class 2, but somewhere there is a cytosolic diversion of endocytosed antigens. From here, they come here and pass through the endosome. So anything which will pass through the proteasomal machinery will get. So that's what we should remember. So don't get confused. So the cross presentation are normally confusing, but don't get confused because anything, all proteins or peptides, even if they are taken up from the outside, if they pass through the proteasome, they will get loaded because that's the channel they find. So again, I come back to this. This is where the antigen activation takes place, and that's how it happens. You have the second signal, you have the third signal. We repeat this. So now, this antigen presenting cell, we all know, has got all the machinery as well as the capability to present the antigen and activate. But under many circumstances, when it is in the resting stage, the co-stimulatory molecules are not expressed. As I said, the immature dendritic cell, if it happens to interact with the antigen-specific T cell, then it leads to no response or tolerogenic response rather than activation response. But once the activation occurs, it expresses all those second signal molecules, the co-stimulatory molecules, and you have the T cell activation. So that is mandatory, it happens. So dendritic cell, immature cells, they cause tolerance. So I told you in the clinical conditions, in the disease conditions, this is one way of, this thing. So then there are certain, co-inhibitory molecules also, because this is natural. If there is antigen activation, T cell activation, it has to be curbed. So naturally there are certain regulatory molecules, or they are co-inhibitory molecules, which get expressed, and most of them are here. But pathogens, they take use of these to modulate the overexpression of these, so that they do not, they make the T cell inactive. So it doesn't go into activation cycle. That's how tumors, they do it. And that's where the immunotherapy has come, which because these molecules, they put a brake on T cell action, and if you use the antibodies against them, you remove the brake, and the cell again starts working, and it happens. So that happens. So these are PD-1 and all that. It happens. So both PD-1, CTLA-4, they act in a different manner, because PD-1 activates certain phosphatases, which dephosphorylates all these, signal transduction molecules, whereas the CTLA-4 has got 4 times more affinity towards the same molecule. So both of them, they basically act differently at different stages. This CTLA-4 acts at a priming stage when the cell is being primed to action, and this the effector stage. So both mechanisms are different. That's why the combination therapies have become more successful than the single therapy, and that's true for everything. So I'm going to summarize whatever we have said. What we have seen is there's an antigen presenting cell which has the capacity to process the antigen and display it on the surface in conjunction with MHC1 or MHC2. If it is MHC1, it activates CD8 cells, which further differentiates. And if it is MHC2, it activates CD4 cell, which further goes and activates other cells. So then there is T cell polarization, which occurs because of the cytokine milieu which is come there. So I think I'll stop here. And thank you very much for your kind attention. You can always ask me questions.
Can one predict exactly what the peptides are that will be, resulting from proteasomal degradation and presented?
Sir, कह रहे हैं कि एक protein है expressed in the cell. Can somebody predict which particular peptide will be loaded and presented, right?
Yeah.
Onto the
how it will be digested by the proteasome. Because it doesn't give all possible peptides.
Yeah. So if it is expressed inside, then it goes through that proteasomal machinery, breaks it up. So there are HLA which have specificity for certain antigens.
No, is there any specificity coming at the level of the proteasome, including the peptide?
मुझे पता नहीं सर, I don't know. होगी तो मुझे नहीं पता, but I'm sorry about this. I'm not a protein
Like for
chemist, but
DNA, we have restriction enzyme for particular sequences. No?
you can generate that because if you can isolate, I mean, I know that we can make MHC tetramers and do all that and specific, but if you can predict which one will be there, so I don't know if it is possible. If it is there, I don't know. Maybe I'm ignorant about it.
Of
Sorry.
course, not tools available in silico where you can predict based on the score. But what Arvind is asking very differently. Just like restriction enzyme, 6 nucleotides have to recognize whether proteasome has something like that. It's not very clear, but
Vinita, can you
some evidence actually, some reports suggest there are certain hydrophobic amino acids. While proteasomes are walking through, they sense. And that seems to be giving them a signal that, oh, this is the peptide once it generated. So they are the ones,
So there must be,
and
because
then trimming happens, this is where particularly for, for the CD8, at least epitopes, So if it is a longer peptide, then they start trimming and then create these.
so trimming. In fact, I deliberately missed, but that is done to fine tune. That is all right. But I don't know whether that answers your question or not. But very much, thank you very much, Dr. Sarath, because God made some wise heads and others He covered with the hair, right? So me and he. So he and me can think better than. All right, thank you so much for helping me. But any more questions from the? Oh.
So I have one question. So you said that basically the degradation will happen through proteasome complex, right? What if the antigen that is there is non-peptide? Like for example, it's a lipid molecule. Then how do we break it down and then present it? Because as such, what sort of interactions would be there between the MHC and the non-peptide antigens which are there?
Very good. So I was thinking that nobody should ask that question. Presentation of lipid antigens and carbohydrate antigens, they are different from this. So we covered the peptide antigens. The lipid antigens are taken up by different phenomena and they are present CD1A type mechanism and they are, so that's a different channel. Similarly, carbohydrate or polysaccharides, they are taken up by the B cells can do that. They can engulf all that. So deliberately avoided, they say there's a whole process and we can always deal with that, but that will basically take a little more time. We can discuss about it. So lipids and carbohydrates, they are non-peptide. They will definitely have their own way of, even nucleic acids, because you have anti-DNA antibodies, anti-RNA antibodies. So everything is antigenic and they all generate immune response, but their processing and everything is different pathways. So we can talk about it, but it will not be appropriate because the time will not be really.
I have one more question. So basically, you also said that MHC1 will be present on all nucleated cells, right? So I had a little doubt. What about the RBCs? They lose their nucleus after a certain time. So what about
RBCs, mature RBCs, they do not have nucleus, so they don't express it.
Okay.
Well, that is good. Okay. So platelet, whatever will express, they will have MHC class 1 nucleated cells. Others, they don't have it, right? So that's okay.
Thank you, sir.
It's a very great talk. I would just like to know, continuing her question, she said that what about lipid and carbohydrate antigens? But talking about antigens to be immunogenic, we need some amount of peptide. So would not be carbohydrates and lipid individual as an antigens and not immunogenic. They need to be conjugated with some carrier proteins to be immunogenic first, and then they will undergo any exogenous or endogenous pathway.
I mean, you can talk about an artificial situation where you want to make them immunogenic and do that, but cells, they don't make that effort. They handle them separately, and they have separate pathways for them.
So the carbohydrates and lipids would be individually immunogenic even when they are not conjugated with any
No, normally
carrier
they
protein.
don't have separately, but they are attached to the peptide also. But then they do generate immune, like lipophilic cell wall of bacteria, mycobacterium is poorly immunogenic, but still now it is known that it can generate immune response, so handled separately, right?
Okay, sir.
So the question with regard to polysaccharide presentation, uh, it- typically there are two ways. One is making a glycoconjugate, as was asked. The other aspect was whether polysaccharides can be presented. Okay, so there are two examples that have been described. One of them happens to be from Streptococcus pneumoniae, class 1. These polysaccharides are taken up and they are actually processed chemically, not using these proteases. I think it is nitric oxide that is being used to cleave these into smaller peptides, and they are actually presented in the context of MHC. Though this is a cell paper which came, several years ago. So there is another example of the same kind from I think Bacteroides fragilis or something like that. So two examples where polysaccharides can be processed and presented directly. With regard to the glycoconjugate business, what happens is that the peptides, protein conjugates are processed, and in fact, in the MHC groove, it's you chop it up into peptides which include the glycan part of it. The peptide binds to the groove, and the glycan is seen by the T cells in the context of MSC. So that's how it works. Yeah.
So you talked about inhibitory receptors. So, sir, in
Which
what
receptors?
inhibitory receptors?
Inhibitory, okay.
Yes, sir. Present on APCs. So, sir, in what cases is that required? Like, will it give an additional advantage towards self and non-self? Or in what cases it is required? Because usually we want an immune response when a foreign antigen or a
We
peptide.
do need them.
Yes.
So once a cell is activated, it needs to be stopped. The continuous activation of cell may lead to autoimmune disease or more degradation, which is not required. So it has to be regulated. So these are basically regulatory molecules which will be expressed once we have, immune response has done its job. But sometimes these molecules are manipulated by the pathogens or tumors for overexpressed, and they block the antigen activation, this T cell activation process. But otherwise, all these regulatory mechanisms, they are inbuilt process of regulating the activation of immune response. We do not want continuous activation.
Yes. So another question was there. So cross presentation. So sir, we know that proteasomal complexes are present in the cytoplasm, and the exogenous antigens are taken inside the endosomes and then lysosomes. So sir, how is it that proteasomal complex is able to act on that? Like, is it that the peptides come out of the
Cross presentation process, people have still working. They are not how these peptides which are taken into endosome are released in the cytosol and go into that process. Exactly not clear, but that's how they speculate that this is the way they are cross-presented. This is one way. There are cytosolic pathway, there is vacuolar pathway. So there are two pathways how they can go. So people just have hypothesized that one of these pathways may work where these antigens are
Thank you,
gone
sir.
into this.
Good afternoon, sir. Sir, I have one question bit different from your presentations about the MHC class 2. So it's at three loci DP, DQ and DR, and as per I know the DQ is associated with the specific peptide of the pathogen and the foreign particles. So DP and DR plays the same role over here, or it had some different role than the DQ? What do you think, sir?
Yeah, I mean, this question is okay. It's a bit more elaborate. We will have to do it. So if we go into this kind of core, it will take longer time. We'll discuss about it. We'll discuss outside, and I'll talk to you about it.
Okay, sir. Thank you.
DP, DPR.
Hello, sir. I have question that RBC does not contain nucleus. But what about in their early stage when they contain nucleus? So they contain MSC class 1 or not?
But the mature RBCs, they never have nucleus, right?
Yeah, but when
So
they are
they're
in
in circulation. So I don't know whether they have. Yes, theoretically it should be okay. Yes. They lose it, right? So 5% of them, when they have nucleus, they do have MHC class 1. But once they have, they become mature RBCs, they lose. Okay? So good.
Do you have more questions? So thank you so much, Sunil, for wonderful talk and making everyone laugh and make them understand as well. So this is the art, not everyone has it. So thank you so much. Okay. So, okay. which he wanted out of place. Okay. Yeah, just once.
I would like to thank you, sir, for this wonderful lecture. And I request Dr. Arvind to present the token of appreciation to Dr. Sunil. Thank you, sir.
Yeah, just want to add something. So I'm sorry, it was not clear to me whether I should present a very basics of complement or my work. So I kept on talking about what now I see is that basics of complement. So I want to just add, and I'll keep adding as the lectures go on, is that one thing I want to let all the students, which most of the textbooks would not tell you, is that when the complement system was discovered, right? Do you know who discovered it? Bordet? Anybody heard? Okay. Bordet did not describe that as a complement system. Do you know that? So he used the word alexin, right? And it was the Paul Ehrlich who actually described that as the complement. You know why? Because it complements, right, the antibody. So the question is, do you think that the complement also complements the antigen presentation? The answer is yes.
Yes.
So if you take any antigen and I said C3 gets covalently linked, right, then you present, you have much better antigen presentation. The pathway and the details are not known, so maybe one of you can work on it and figure it out. Okay. That's another thing which is there. So on the B cells, what he is referring to, so you heard B cell receptor, right? There is also a complement receptor called CR2. So if you have an antigen linked to a part of C3b molecule, the puppet which I called C3d, then C3d is a ligand for CR2. Then what happens that it sits on that and you have B cell receptor recognizing antigen and CR2 recognizing C3d. This decreases the threshold of the B cell stimulation, so you can give less antigen and still get a response. A wonderful experiment was done almost 20 years now, came in Cell. What they did is that they took hen egg lysozyme, HEL, and they coupled with the C3d, right? They made 1, 2, or 3 copies of it, and without adjuvant, injected. So HEL would not give you so much response, but if you give these copies attached, it enhanced 1,000-fold the antibody response. What it meant was that C3d attached to antigen can act as a molecular adjuvant, right? And the thing is, what he is referring to is because now we know that we have BCR and CR2, they are doing. It also helps in other way. It also helps in retention of the antigen on DCs, right? So CR2 is also present on DCs. It helps in, so if you do not have, for example, complement or C3 knockout mice, your response is only 50%. Rest everything 50% comes from complement. So we need to break for lunch and then we'll be back. So thank you very much, everyone.
The lunch is at the same venue as yesterday.